Welcome to Beyond the Lab, a podcast where we talk about people, ideas, and insights in the science industry.
Okay, sure.
Okay. So with us today is an assistant professor in drug delivery and nanomedicine. He is also a lecturer in the Kuliah of Pharmacy in International Islamic University Malaysia Kuantan campus. Additionally, he was a fellow in Maybank as part of the CEO at faculty program organized by MOE. So hi Izzat, how are you doing?
Hi, Julian. Yeah, I'm great.
Okay, so just as like a introduction to you, right? I'm very interested in that fellow in Maybank thing. Maybe just talk us through a little bit about what you did there as an academician somehow.
Okay, thanks, Julian. So I think a lot of questions people asking me how is it I'm from like engineering come pharmacy and suddenly in Maybank.
Yeah.
Like totally a different kind of like environment, banking, financial, and so on. So basically, like as you mentioned, like there's a program organized by Ministry of Education, Higher Education, which we call it as CEO at Faculty Program 2.0. So this 2.0, so they have the 1.0. Basically, the program is like an engagement between the industry and corporate world, and also with academia. So with the 1.0, they bring the people from industry and corporate to actually work in academia, and they experience and share their expertise and how to improve the academia part. And now on the 2.0, so they bring the academia, people from academia, to actually experience how actually work in an industry and also in corporate. So when we talk about it, people might thought it's just like industrial attachment or something, but it's exactly like it's more on how you learned the leadership and the management in corporate and industry. So not something that you will experience as a lecturer or academic in university. So you want to see how these particular people of corporate organized, like some of them are actually like global level of organization. For example, myself, I'm attached as a fellow with Maybank. So in Maybank, all the fellows, there are 4 of us, so I'm one of them. We attach under what we call it Group Human Capital, or AKA is the HR of Maybank. So I under the chief group, Datuk Nur Ramanaf. So we will experience and how exactly the human capital HR of Maybank, how they do the talent development, or talent management of the staff. Especially surprisingly, when I enter as a fellow in Maybank, so they thought that, so I have this kind of like skeptical thinking, corporate bank, all banking people, financial people, and so on, right?
Understand.
And then, surprisingly, like the one that I'm engaged with, she's actually a person from biotech people. And
is that Miss Sophia?
Yeah, Sophia, yes. You know
her? Yeah, I heard about her,
Yeah, so I was like, wow, people from biotech and head of talent attraction and work futurization of banking company Maybank from biotech. So like, that's where I learned like, exactly how banking is not like what the stereotyping people thought about. And then it's really great. Like academics know how exactly like you deal with people from many backgrounds, and how exactly you can prepare students to actually prepare themselves to work in industry. Because most of the lecturers actually they only know on theoretical, so we do have academics that are from industry, but then it's very small numbers. So most of the academics, they are like freshly from PhDs from the research and they become academic. So whatever they learn about industry, whatever they heard from their friends, or they learn by theoretical. So this is for me, that's really brilliant movement by the ministry. Yeah.
So that's a little bit about Izzat and a little bit about the PhD aspect of things. So today's topic is nanomedicine. So when I hear about nanotech, right, it's very sci-fi, very tech. It's like something that I won't enjoy when I die. But in a health context, right, and you deal a lot with nanotech in your research, right? What is nanotechnology?
Okay, so nanotechnology is another essential science technology terms. Like we use a nanomaterial. So basically, when you talk about nano, it's a very small size. Okay, I think if I want to give a simple like micro, so micro like a size of a bacteria.
Yeah.
So bacteria is like micro size. Nano is a virus size. So much smaller. So we are trying to use many type of material. Like for example, we have metal base, we have organic base, non-organic base, even we have lipid base of material. We modified them and then we make into a nano size of 10⁻⁹. And then we use that particular material kind of like a car. So we make a very nano-sized car, and then we try to deliver drugs or any therapeutical materials to cure diseases, or even to do diagnostic, when you want to screen where are diseases located in our body system.
Okay, cool. So before we dive into further about nanotech and what's your research, maybe just one or two liner, tell us like what has your research been about so far?
Okay, so my research, if people browse my website, they will say I would say my expertise in nanomedicine, drug delivery, protein engineering, genetic engineering, and also in oncology. So mainly, since my undergraduate in engineering and my PhD in pharmacy, I'm working on we develop materials, nanomaterials to become carrier to actually cure cancer. I mean, to therapeutic or even to diagnose cancer cell, cancer tissue are located. So that's the main of my research. We are not developing new drugs. We're not finding new drugs. We try to improve what available drugs available in clinical. And then we try to improve, because we know that, for example, chemotherapy, like most certain people got fatality not because of the cancer, because of the drugs. So the drugs really work to kill cancer, but then sometimes it kills you before it kills cancer.
Oh yeah.
Because of the side effect. Because you inject into the human body and the drugs is just go everywhere. So we want to avoid that. Yeah, it kills everything. We want to avoid. We know it works to kill cancer, but we don't want to kill the host. The purpose we want to save the patient, right? So we try to reduce as much as possible of the side effects. So then we can, like a car, you just bring your passenger like a Grab, right? You just bring your passenger to destination. That's what we're going to do. We just bring the drugs to our destination, which is the cancer cells, and then allow the drugs to do the killing, or even to locate where exactly the cancers are in our body system.
So I think the term for this, and correct me if I'm wrong, is precision medicine, right? Is that the term for it?
Yes, it's precision. So nowadays, people know as precision medicine. Yeah.
Yeah. So your research, you deal a lot with hepatitis B virus. Maybe just tell us a little bit about that and what's so unique about this virus and why do you choose to work with it?
Yeah, so I started working in nanomedicine with hepatitis B virus. So we know the virus itself, it's really easily to infect our body system. So we know it works like that. But I said, in other way, how about we use that same property, we modify them, and then we use the property of it could infect the human cell instead of it deliver and cause disease. We remove all the viral genome or DNA, RNA involved in it, and then we replace that with drug. So we see that potential of that Hep B virus can deliver drugs. So in my case, I modified the virus. So we only take the middle of the protein, like we call that core particles or core protein, which actually that's the one who delivered the DNA to cause the disease. So we remove the DNA inside, just the protein, and then we know the protein can easily penetrate to cells. And then we deliver, in my case, because I'm doing genetic engineering for my undergrad until my PhD. So we add what we call it small interfering RNAs. So this RNA, we call siRNA or siRNA, have a function that you can't block whatever the RNA that can produce protein that causing cancer development or cancer growth and so on. With that, the property of that Hep B easily infect our tissue cells. So I say we use this property instead of we thought it's a bad, we change that into become positive effect, and then we want it to specific target HER2 cancer cells. So HER2 is one of the receptors that are available in many types of cancer, mainly in breast cancer and ovarian cancer. We want that it's only deliver the siRNA to HER2 express cancer cells, and then it was the killing. In my case, I do genetic engineering. So for that, we try to block or inhibit the growth of the cancer cells, so that when we stop the development of the growth of the cancer cells, so eventually you will cause it to die.
Okay, so maybe let's dive a little bit into siRNA, like siRNA.
Okay.
Is that how you pronounce it?
siRNA, yes. Yeah.
Yeah. So let's dive a little bit about that and tell us how does siRNA actually blocks the gene of HER2 cancer cells?
Okay. So basically, if you talk before we go with siRNA, we talk about how the protein works in our body, how being protein produced.
Sure.
So basically, we have a system that we have the translation and translation. I mean, we call it the dogma, the protein dogma, and so on.
Yep.
So basically, our DNA will be translated and be transcribed, and then we have RNA. RNA will be translated, and that will produce protein. So it depends whatever the information that the RNA being taken off and translated to protein, it will contribute into certain things. So in this case, we know that certain RNA or actually causing the cancer growth. So this kind of beta of RNA or ribonucleic acid that causing the production of protein that promote the growth of the cancer. So small interference RNA or siRNA has the function to block the RNA being translated. So it functions as like you kind of like a police block someone in a roadblock. So this kind of so that's kind of
a good
police block
analogy,
that so that the car will not go further. So in our case, certain will block from the RNA being translated. So when there's no protein to promote the growth, in my case, like the growth of the cancer, so then it will not causing the cancer to keep growing and then causing it to die because they don't receive the information. So certain can block that information to be before being translated.
That's a really, really good explanation. And your research focuses on cancer therapy. So maybe just to compare, right?
Yes, both cancer therapy and cancer diagnosis.
Just to compared to like traditional cancer therapy, which is chemotherapy, I believe. Yeah, and I know we talked about this earlier a little bit. Maybe just compare the key differences between like just the chemotherapy and your method. Like how is yours superior?
So chemotherapy, like they're using the chemotherapy or anti-cancer drugs. So basically, each drug has their own function to kill the cancer cells and depends on type of cancer cells. And then whatever what called the pro-drugs being used in clinical, they're being injected or being given oral, depends on the cancer. And then the drugs will do the work to kill the cancer. But however, as I mentioned earlier, because there's no specific vehicle to bring the drugs. So the drugs goes into our body and then it depends on our immune system, depending on how strong our body is. Like the drug will do the killing while arrive at the target. So like, for example, if you inject it to the bloodstream, so throughout when it goes to the bloodstream before, for example, like you have like colon cancer, because it reaches the colon cancer area, it will do the killing bit by bit by bit by bit. So that's why we see all these side effects. People got bald, lose appetite, and sometimes people got on it, and etc. etc. So that's the problem with the conventional chemotherapy approach. With our case, we know the chemotherapy has very great potential to do the cell killing. But then we know the side effects, as I mentioned earlier. So we say, why don't we develop a special vehicle that only bring the drugs to only the targeted cancer cells, in our case, like in the tumor area or any cancerous spot. So with my approach, we have a special vehicle for those drugs, and we just bring them, and then we modify also the surface of the vehicle so that it only recognize specific cancers. So then theoretically, it will only reach the cancer area or targeted area, and then it will release the drug, and the drug will do killing on that particular area. So you will avoid as much as possible being in contact with healthy tissues, and then you will not do the killing with our healthy tissues and cells. So so that's kind of superior. Like you have a specific targeting, and and another thing, we can lower the dosage, because like the reason, another thing how the killings happen, for the drugs to the conventional one, for the drugs to actually arrive to the cancer cells, only a small percentage. So for them to actually meet that small percentage, they put a high dosage. So that's also causing the really huge side effects. But for us, we know only this amount of drug needed. So why we just deliver this amount rather than we put an extra, because you know the percentage will arrive just small. So that's the superior of this nanomedicine approach.
So basically lower dosage and you don't kill as much, right? And it's more precise.
That's always people in nanomedicine feel. Low dosage, high efficacy, and safe.
Okay. So I think I want to expand a little bit on the targeting pathway, if that's all right with you. How does the nano carrier, or in your case virus, know how to target specifically the cancer cell?
Okay, so now we talk about the specific targeting part. Okay, like in terms of administration of drugs, or I think for those in medical or pharmacy that you might from there, we have many administration routes. We have intravenous, through nasal, sometimes through oral. So I think I will not touch that part. But then the question is, as you ask, like how they know what kind of data we give to that vehicle that recognize that. For example, you take MRT, the ticket will stop there, right? Like, you put the grab, you put the you arrive there, instead of like you want to go to 1U, it send you to the gardens. No, it will go to 1U, will not go out there. So how exactly we give that information to the vehicle? So basically, you give example like I said, the vehicle is kind of like a ball, right? So inside the ball is all the drugs. But then if just that kind of vehicle and there's no other information, then it will just go rolling all over our body and then doesn't go anywhere. So we need something to recognize, like a particular information to arrive to a particular area. We take like each cancer has their own what we call the biomarkers. So in other simple or layman term, they have their own ID. Like each cancer have their own ID. So we need to have information to recognize that ID or biomarkers. we modified the surface of the nanocarriers or the vehicle. Like we have like what we call it a targeting moieties or epitope that only recognize that biomarker specifically. So until it reach to that biomarkers, it will not send a signal to the ball to release whatever inside. So how exactly it can track? Like it will search. So that information that you have, like if you know how the COVID-19 virus look like the spike, so we modify actually our vehicle also that kind of spike. So as much as possible of the spike. So that spike kind of like will grab, this is the actual biomarkers that will react. If that's not the ID they can recognize, they will not react. So that's how exactly when you inject any nanocarriers or any nano vehicles that deal with drugs or to scan, it based on that the targeting moieties that on the surface that you will only recognize specific biomarkers or ID of the cancers.
So you explored a little bit about sRNA just now. What other drugs what other drugs can we actually use to target cancer cells? Like what other drugs have you explored personally?
With the nanomedicine, interestingly, like you are talking like a very universal carrier. You can put anything inside of it, not just sRNA. You can put protein, you can put DNA, you can put plasmid, you can even put anti-cancer drug. So like I've been working, for example, few types of anti-cancer drug like doxorubicin, we have DTS, and then right now I'm working with my project in Malaysia, we call it fluorosil. So we are targeting that with oral cancer, and then we also a few more drugs right now. So those are you can encapsulate, or we got the term encapsulate to put it inside the vehicle. So actually, whatever you want to put, you can put it. For example, like if you're not sending for therapy, not drugs, but you want to know where is your cancer. Like for example, like the non-tumorous cancer, or like you have a breast cancer, but then it's already like stage 3 or stage 4, then it's metastasized. But then you don't know where exactly the cancer, especially if not non-tumor. So we can send kind of like a fluorescent marker, or like for example something to actually attach, and you can track them like a trackers. You can put trackers with your vehicle like inside of it, and then it will release it will bounce to your cancer cells, and then you give a signal. So there are a few types of trackers. You can put that fluorescent, or you can put we call luminescent, another type of protein, or the one that I really like to work with, we put radionuclide or the radioactive material. So it can track once it's bound to the cancers. You can live imaging and track where exactly your cancer in your body. So I worked that for my PhD before. We tried to track with the HER2. We inject in the animal, in the mice, and we only know that we do like live imaging 24 hours with the animal, and then we know that our vehicles, in my case, my virus, my core protein, only attach and localize at the cancer cells, at the cancer tissues. So the good thing for diagnosis, I mean, they talk about diagnosis, like you will know, like the doctor know, okay, so basically your cancer is here and there and there. So actually, people want to know after post-surgery, for example, like you remove the tumor and so on, just to ensure whether you still have cancer cells in your body or cancer tissues, that when you inject again with this nano carrier with the trackers, actually, when you see there's nothing more left there's no nano carriers can be traced, that means nothing left. But there's some accumulation here and there, okay, we are not done here, guys.
Understand. That's good. So your recent research has been more on that, right? The biomarkers and allowing people to know where their cancer is. So what's next for your upcoming few researchers in the next few years?
So the next few years, of course, like we want it to be clinical. So we want to make it into product to commercialize. But of course, there are a lot of challenge into like you go to clinical stage 1, stage 2, stage 3. So there are quite challenge. So the next stage actually like whether we can scale up our formulation and then can be tested on the clinical. So that's really the next stage for this research. And then another thing with the nano, I mean, if you talk about Malaysia, the demand is still low for the nanomedicine or precision medicine. So the cost actually is more than conventional chemotherapy.
Okay,
but conventional chemotherapy is already expensive. Then we talk about nano, like they say like sci-fi, like it's much expensive. So our target is how can we deliver, like for example, for producing cancer using our technology at a very, very low cost. It's like we want to avoid all these expensive, we want that it's accessible for many people from many various backgrounds. So we need to think, okay, you want to deliver this, that we need to come with a very cheap material, very less cost, and then but still have not just a good efficacy, perhaps much better than the current chemotherapy approach.
So basically, right now the efficiency of producing the virus is very low, right? Because I did it in uni back then also. How do you actually make it faster? Or how do you actually make it low cost? I'm just curious about it. What's your plan
Okay,
for that
so
part?
the low cost actually like the material first. The material, like for example, like we are working with the biological material previously. Like we know like to make it commercialized is still a long way to go because like the limitation of technology to production of the protein and then the whole steps. So we need to re-engineer back again in terms of that the production of the protein. So right now we try to find an approach from a different low cost material. Like for example, I'm working with plant-based material as well as the vehicle and also the polymer based. So like the plant-based that now I'm working with like the one that you consume as your dessert, your jelly. All this agar-agar or in Malay we call it, right? So people know that's for food to make dessert. We said no, now we explore that particular material for medicine. So it's really cheap. Like you buy all the material to make your jelly. It's like how many ringgit?
Yeah.
Right. So we say like the sauce is very cheap, and we have like not unlimited, but like quite abundance, like the seaweed all over in the sea and so on. So we try to explore on that material as well. So like the drug itself is already expensive. So from the business point of view, for you to have a very lower cost in terms of treatment of chemotherapy, I think the reason it's expensive because you need to put high dosage, right, to the patients. So if you cut the dosage, so perhaps you cut about 1/3, even quarter of the cost, and then you're using cheaper material, and then there's no need much of like times of you need to be injected, and then you will see that the whole cost will be cutting down, and then the more demand from the patient, and then the more supply will provide, and then you cut further costs in terms of like that.
Economies of scale, right?
Treatment. Yeah.
So are there actually, just curious, right? Are there actually any nanotechnology out there in the health world?
In Malaysia, not yet much that being in clinical, but then in states, in Singapore, they do. There are many types of like nanomedicine approved in the FDA. They already not just in clinical, but in hospital have been use it. So they're quite numbers in states in other part of the country, but not in Malaysia. I think like the demand is not there, and then people are not really realize like actually we have other options. But for immunotherapy, we do already started in Malaysia, but then the cost, I already asked my friend work in that part, like it's for those who eat 20 people only. So like really like for the high class people, like for those like M40 and below, like you can just watch and waiting for some miracle happen.
Klinik kesihatan.
Yeah. So
how about Singapore? Is the cost high there?
Singapore is quite high. I think Singapore is quite high actually in terms of like, but they do starting with the nanomedicine. Like we know that one of the professor from NUS, the Chinese lady. So she also like said one of the is already going to clinical stage right now. So and then she also tried, even like last talk I attend about with her, she also tried to approach like, okay, we need to find like a cheaper cost. I think the whole thing that cost is very expensive, not just the drug but the production cost. The step or to produce. So for example, like I always say to my colleagues or any scientists or researchers out there, actually do working in nanomedicine, like when you try to come a new idea, don't think up to lab scale. Think of the end product. You want it to be on the pharmacy, you want to be available. Right. So you need to think that's your target. So you need to start from the beginning. Okay, I need to find something so that it can be there and can be accessed by anyone.
But we've got to think like 5 steps ahead, right?
Yeah, so you need to think 5 steps ahead. So like that's what my supervisor taught me when I was doing my PhD. She thought, okay, Izzat, you need to think until the animal level. I mean, preclinical stage. But when I came to Malaysia, one of my mentor, which actually Dr. Amani, who's lead the COVID-19 kit right now, she told me, now Izzat, you need to go further step. Think about the end product in the hospital. Okay, so I did like, okay, I need to change my whole plan. Yeah, furthermore.
Aside from cancer therapy, right, what else can nanotech be applied in?
Okay, that's interesting. It's like because it's, you can modify your nanomaterial, your nanocarriers to any disease that you want to target. Okay, so it's not just cancer. Like if you, for example, like you think like for cardiovascular disease that you want to deliver the drugs, so it's best you just go to the cardiac tissues. So you just add the cardiac drugs, and then you modify the surface that only have recognized the biomarkers on cardiac tissues. So you can deliver on that. So
very flexible.
Because I'm working with the COVID-19 as well. So right now I'm working with for the diagnosis kit, which currently we are competing with MIT Soft Challenge. So That's why for diagnosis, but then we see the potential that if there is any antiviral drugs that work, we can use the same approach to deliver to kill virus. So rather than the antiviral drugs go everywhere, try to find its luck to find where the COVID virus located is, how about you just bound specific our carrier, just go bound to the COVID-19 virus, and then the drugs just deliver to the virus instead of just go everywhere like what happened with chemotherapy. So with the nanocarriers or nanomedicine approach, they're kind of infinity potential. You modify them, you understand the markers, the biomarkers, the ID, whatever tissue you want to to localize, then you can do all the work wherever you want to deliver.
I actually came across that competition on your Facebook page. Maybe tell us a little bit about that and what's your progress in that stage.
The kit.
Yeah, the MIT.
Okay, so basically, let me rephrase by all the kits. So basically, leading by Dr. Amani Salim, and also I'm also one of the leading scientists for the project. We came out the idea of the project, like why don't we came a diagnostic kit that non-invasive for COVID-19? So we know, like not just in Malaysia, all over the world, the best approach to screen whether you are positive or negative COVID is through RT-PCR. So basically, they need to have do I do the swab testing, go through the nose, go through the mouth, and take the back mucus at the back and do some screening. And then for those who are familiar with RT-PCR, the whole process costs really a lot. For example, in Malaysia, like if you go to private, the private give you the actual cost about 500 to 600 ringgit per test because the reagent is expensive. I mean, the government is free because we are subsidized. But then for those like can't wait, want to read, or that you are not have access for the government subsidy, so you need to go to private because like the whole machine itself like 100,000s, and then the reagent is another story. And the limitation with RT-PCR is the possibility of 30% of false negative or false positive. But we really scared. False positive is fine, so at least, but it's false negative. You thought you're negative, but actually you're positive.
Spread to everybody.
Go spread everybody like a welcome COVID, like something like that. So we see that there's something we need to do. First, and it's so expensive, and then it's inaccessible for everyone. And if you talk about not just Malaysia, in the other part of the world, like for the African country, for the very poor country, like they can't have that access to know whether they have positive or not. So we say, we need to sit down and came out with a non-invasive chip, and then everyone can use it. So because another thing with the swab testing, it must be done as a specific diagnostic lab, and you need a lot of frontliners to work on it. And you know how frontliners have been struggled since the beginning of MCO. So we see, there's so many problems that we need to tackle, help the country, and not just country, to global itself. We came out like the idea we need to combine the nanotechnology from the engineering and the nanomedicine people, and also from the health people, came up with something, a kit that everyone can access, and then it's not invasive, so there's no need swab testing. So our kit, we came out like with using a concept from a glucose meter. So glucose meter that have like you use your saliva and give you information whether you're high glucose or not. We use that kind of concept. We modified, we make a special sensor, which actually the ideas also came from Dr. Amani herself, that she's a same concept that she used the sensor when she involved in the project of a NASA satellite before. So a same concept we put in our kit, and then it will bound so we use the nanotechnology, which we use a nanoparticle. In our case, we use a few types of nanometals between silver and gold, and they have specific antibody that only bound to SARS-CoV-2, which is the COVID-19 virus, and that it will give you information whether you're positive or negative less than 5 minutes with accuracy more than 95%.
5 minutes, wow, that's fast.
So with the RT-PCR, the quickest you can get is 6 hours. The quickest. It's 6 hours not before starting off your swabbing, starting the sample arrive at the lab. Okay.
Understand. The interval between the swab to the lab also takes it'll take them.
That's true. That's another story. So like in our case, like we want to 5 minutes and just using saliva, and then you put a saliva on the kit, and they will give you positive or negative within 5 minutes. And then that's the whole project. So we are competing with a competition organized by Massachusetts Institute of Technology, or MIT. So they have a competition, they call it MIT Soft 2020 Global Challenge. So they call all of the solutions, researchers, industry people to come out with solutions to settle the world issues. And we in under categories of health security and pandemic. So we came out with this project proposal, come to them, and then it was like about 736 solutions from 95 countries for that category. And then we didn't expect we've been finalized top 15. The only team from Malaysia. The only team from Malaysia.
Congrats. That's
so
huge.
right now, in terms of progress, we are doing further. So we done all the in silico studies. So that's why our information is 95% accuracy, and then within the 5 minutes, and then the limitation of detecting the virus is enough. You only have 5 virus in your saliva, in 1 microliter of your saliva. If you only have 5 virus, we can detect already. You don't need abundance of virus, just 5 virus. That virus is nano size, like 5 virus, like still you thought there's nothing in your saliva.
Yeah. So how close is
the whole project? Yep.
How close is that to commercializing? Or is it already commercialized?
So it's not yet commercialized.
Okay.
So that's why we're competing with the competition, because we need fund. We need fund for that, because like our project, we are not doing the manufacturing. We came out we kind of like an architect. We made the prototype, we made the design, and then we're going to ask for those have big pharma company or the investors interested, so that we can produce at very cheap, because we are target for the kit to be commercialized as low as $1 or equal to 5 ringgit per kit. Okay. So instead of you pay 600 ringgit for half day or the next day you can know the results, then you need to do another 3 times to confirm. Then if you have that kit only 5 ringgit, then it's much cheaper than your boba. So if you buy 2 boba, it's enough for you to 4 times or 5 times of testing whether you're positive or not. Target that everyone can do the checking at home. When you feel like, oh my God, am I got COVID? You just have the kit, put your saliva and know the information. So you don't have to queue in to the hospital, and then there's no need to have special diagnostic lab, and you don't burden the frontliners anymore. So I think the whole cost thing, the country have been haywire with the funding and financial, because it took a lot from the whole testing. And then there's been cost, burdening the whole country for that. So that's where you're coming up. We're not settled just helping for assessing whether you're positive or negative of the COVID. We're also helping not just our country, any countries, enough for burdening yourself with all this expensive kit.
So that's good. I think throughout this episode, we can really learn that, nanotech is the future, and it's definitely better than the traditional methods.
Yeah.
And so
that's why
throughout your research,
hopefully people out there, please don't say goodbye to science and technology. Please come back, and then we have this nanotech. It's not sci-fi anymore. It's not like Star Wars story anymore. It's already there. And we need more people to join us, and then we bring to another level after this.
Okay, so throughout your research, just for a last question, what's your best key takeaway?
As a researcher.
As a researcher.
Okay, as a researcher, I realized that there's so many uncertainties happen in the world. Especially you cannot say you are a researcher if you're not find yourself facing failure. That's what I learned when I started in the research world. I stuck to make my own plasmid for 2 months. I've been doing the same thing. Why did it get stuck? And then that's give me fun. Okay. And then another thing that really put me real interesting, I would say that sparked myself to continue to become a researcher, that I'm doing this not for myself. I'm doing this to help people out there.
Yeah.
So that always kept me motivation, that whatever I do, if I feel stuck, stay in the lab until 4 a.m., 5 a.m., couldn't find any new things. I always say to motivate myself, I'm doing this to help people out there. I cannot be a medical doctor because I'm scared of human blood. No, I'm scared. I don't scare human blood. I'm scared of surgery. I cannot stand doing surgery to human.
Understandable.
But I have no issue with surgery. I have no issue with surgery of animal. I think because I feel I'm cooking God. So I cannot be on that part. But I feel this is part of my contribution to my research. So regardless whether your research is sciences or social sciences, you know that you're doing this to solve some issues out there. So that kept me motivation. That what put me okay, whenever any struggle that I face in research world, that give me spark and give me motivation to pursue. I know I do this, even I cannot see the solution coming when I seem alive. I know the knowledge will be transferred to others, and then somebody will contribute.
It's effect,
Franklin Wilkins, right? She didn't know her contribution, she died because of that finding DNA, actually helping the whole world.
Yeah, true.
Right? She didn't know, but then that's what happened. So that was that kept me passionate. That gave me to continue my work as researcher.
Okay, so that's all. So thank you so much, Izzat, for joining me today. Hope you enjoyed this.
Yeah, thank you very much, Julian, for the opportunity. I really had fun talking with you, discussing with you.
Yeah, great. It was great. I really learned a lot.
Thank you for tuning in to the Beyond the Lab podcast. I hope you enjoyed this episode as much as I did making it. So stay tuned for the next one.